Hello everyone. We are going to explain about atomic absorption spectroscopy. Introduction. Atomic absorption spectroscopy is very common technique for detecting metals and metalloids in sample. It is very reliable and simple to use. It can analyze over 62 elements. It also measures concentration of metals in the sample. History of atomic absorption spectroscopy is: the first absorption spectrometer was built by CSIRO scientist Alan Walsh in 1954. The elements detected by atomic absorption are highlighted in pink color in given periodic table. Principle behind the atomic absorption spectroscopy is that the technique uses basically the principle that free atoms or gases generated in an atomizer can absorb radiation at specific frequency. Atomic absorption spectroscopy quantifies the absorption of ground state atoms of the gaseous state. The atom absorb ultraviolet or visible light and make transitions to higher electronic energy levels. The analyte concentration is determined from the amount of absorption. Concentration measurements are usually determined from a working curve by calibrating the instrument with standards of unknown concentration. Atomic absorption is a very common technique for detecting metals and metalloids in environmental samples. Instrumentation of atomic absorption spectroscopy includes light source, nebulizer, atomizer, monochromator, and detector. Here given is the diagrammatic representation of atomic absorption spectroscopy. The first one is light source. Hollow cathode lamp is the most common radiation source used in AAS. It contains a tungsten anode and a hollow cylindrical cathode made of the element to be determined. These are sealed in a glass tube filled with inert gas, neon or argon. Each element has its own unique lamp which must be used for the analysis. The first one is hollow cathode lamp. The hollow cathode lamp is an excellent bright line source for most of the elements determinable by atomic absorption. The cathode of the lamp frequently is a hollowed out cylinder of the metal whose spectrum is to be produced. To provide hollow cathode lamp life, some manufacturers produce lamps with largest internal volumes so that a greater supply of field gas at optimum pressure is available. The second light source is electrodeless discharge lamp. A small amount of the metal or salt of the element for which the source is to be used is sealed inside a caused bulb. This bulb is placed inside a small self-contained RF generator or driver. When power is applied to the driver, an RF field is created. The coupled energy will vaporize and excite the atoms inside the bulb, causing them to emit their characteristic spectrum. An accessory power supply is required to operate an EDL. Primary combustion zone. It contains initial decomposition. Primary combustion zone. It involves initial decomposition and it contains the molecular fragments. The temperature of primary combustion zone is cool. Interzonal region is the hottest. Most atomic fragments are present here and it is used for emission or fluorescence. The secondary combustion zone is cooler. Conversion of atoms to stable molecules occurs here. Element rapidly oxidizes, largest atom near to burner, and the element oxidizes poorly is largest atom away from the burner. Most sensitive part of the flame for AAS varies with analyte. The next part of instrumentation is nebulizer. Nebulizers suck up the liquid samples at controlled rate. Create a fine aerosol spray for introduction into the flame. Mix the aerosol and fuel and oxidant thoroughly for introduction into the flame. The second part is atomizer. Elements to be analyzed needs to be in atomic state. Atomization is the process of separating particles into individual molecules and breaking molecules into atoms. This is done by exposing the analyte to high temperatures in a flame and graphite furnace. The atomizers most commonly used nowadays are spectroscopic or flames, and the second is electrothermal, that is graphite tube atomizer. We will see one by one. The first one is flame atomizer. Different flames can be achieved using different mixtures of gases depending on the desired temperature and burning velocity. Some elements can only be converted to atoms at high temperatures. Even at high temperatures, if excess oxygen is present, Some metals form oxides that do not redissociate into atoms. To inhibit their formation, conditions of the flame may be modified to achieve a reducing non-oxidizing flame. The second one is electrothermal or graphite tube atomizer. It uses a graphite coated furnace to vaporize the sample. Samples are deposited in a small graphite coated tube which is then heated to vaporize and atomize the analyte. The graphite tubes are heated using a high current power supply. Steps in electrothermal atomization are drying, pyrolysis, atomization, and cleaning. Monochromator. This is a very important part in atomic absorption spectroscopy. It is used to separate out all of the thousands of lines. A monochromator is used to select the specific wavelength of light which is absorbed by the sample and to exclude other wavelengths. The selection of the specific light allows the determination of the selected element in the presence of others. Detectors. The light selected by the monochromator is directed into a cathode, that is typically a photomultiplier tube, whose function is to convert the light signals into electrical signals proportional to the light intensity. The processing of electric signal is fulfilled by a signal amplifier. The signal could be displayed for readout or further fed into a data station for printout by the requested format. Types of detectors used in atomic absorption spectroscopy are photomultiplier tube, barrier layer cell or photovoltaic cell, phototubes and photoemissive tubes. The diagrams given are of detectors used in atomic absorption spectroscopy.
Single beam atomic absorption spectrometer. In this there is a light source, collimator, monochromator, slit, sample and a detector. In double beam atomic absorption spectrometer, there is a light source, collimator, monochromator, slit, reference and sample. M1 is the beam splitter, M2 and M3 are reflecting mirrors, M4 is the grid mirror and there is a detector. Calibration curve. A calibration curve is used to determine the unknown concentration of an element in a sample. The instrument is calibrated using several solutions of known concentrations. The absorbance of each known solution is measured and then a calibration curve of concentration versus absorbance is plotted. The sample solution is fed into an instrument and the absorbance of the element in the solution is measured. The unknown concentration of the element is then calculated from the calibration curve. Next, we discuss the interferences in atomic absorption spectroscopy. Interferences is a phenomenon that leads to change in intensity of analyte signal in spectroscopy. Interferences fall into two basic categories. First, non-spectral interferences, and second, spectral interferences. The non-spectral interferences affect the formation of analyte atoms, and spectral interferences highlight absorption due to presence of absorbing species. Non-spectral interferences. First, matrix interferences. When a sample is more viscous or has different surface tension than the standard, it results in difference in sample uptake rate due to changes in nebulization efficiency. Such interferences are minimized by matching the matrix composition of standard and sample. Chemical interferences. It is sample contain a species which forms a thermally stable compound with analyte that is not completely decomposed by the flame energy. Then chemical interferences exist. Such interferences are minimized by using higher temperature to provide higher dissociation energy. Next is the ionization interferences. It is more common in hot flames. The dissociation process doesn't stop at formation of ground state atoms. Excess energy of the flame lead to excitation of ground state atoms to ionic state by loss of electrons, thereby resulting in depletion of ground state atoms. Ionization interferences are eliminated by an excess of element which is easily ionized, thereby creating a large number of electrons in the flame and suppressing the ionization of analyte. Next is the spectral interferences. These are caused by radiation overlap of absorption line due to emissions from another element or compound. If an absorbing wavelength of an element not being determined but present in the sample falls within the measuring line of the element of interest, the absorbance of the element will be measured together with the analyte of interest and give a higher absorbance value. For example, vanadium line is 3082.11 Armstrong and aluminium is at 3082.15 Armstrong. Choose a different aluminium line at 3092.7 Armstrong. Note: interference due to overlapping lines is rare in atomic absorption spectroscopy. Spectral interference is also affected by the radiation of an emission line of another element or compound, or background radiation from flame, solvent, or sample. Example 2. Radiation interference in the determination of sodium with magnesium present and in the determination of iron with copper or nickel present. Use alternative wavelength, use smaller slit width, or use a blank. Due to matrix, the matrix contains molecular species which can show molecular spectra. This might result in various spectral interferences such as 1. Overlapping of two lines. Molecular spectra is much broader with a greater net absorbance, so it may overlap with the actual atomic spectra. Second, presence of combustion products. The radiation may scatter due to this particulate matter, and we get less absorption due to atomic species. Third, absorption of radiation by matrix components. Next, we discuss the methods of correction. There are two methods. First, two-line method. For this method, we need to know the absorption wavelength of interfering species. We will choose two wavelengths close together. One wavelength is the absorption wavelength of the analyte, and other wavelength would be the absorption wavelength of the interfering species. But the analyte must not absorb at the wavelength of the interfering species. The interference may absorb at the wavelength of the analyte. Since the two wavelengths are closer, the absorbance and absorptivities will be constant. By comparing the total absorbance at the first wavelength and the absorbance at the second wavelength, we can eliminate the interference due to molecular species. The formula goes A of atom plus molecule minus A of molecule is equal to A of atom. Second is the continuous source method. Deuterium lamps can be used to capture molecular absorbance. Place a hollow cathode lamp and a continuous source deuterium lamp and let the radiations meet and pass through the sample. Hollow cathode lamps can record the absorbance of atoms and molecules. The deuterium lamp will record the absorbance only due to the molecules. Next, we discuss the Zeeman effect. When the gaseous atoms are placed in a magnetic field, they orient themselves in the direction of the magnetic field. But this orientation is not seen for molecular species, so the electronic energy levels get split. This is called the Zeeman effect, which can be used to remove background from the spectra. Applications: determination of small amount of metals such as lead, mercury, and magnesium. It is widely it is widely used in metallurgy, alloys, and in inorganic analysis. Determination of calcium, magnesium, sodium, and potassium in blood serum. Biochemical analysis. Determination of metallic elements in food and industry. Environmental studies and pharmaceutical industry. Advantages: accuracy. Atomic absorption spectroscopy is highly accurate method that can produce results with a rate of 0.5 to 5%. Sensitivity. It can detect metallic elements in small quantities down to parts per billion. Cost. It is often uses less argon than other methods, so running costs are often lower. Accessibility. Atomic absorption spectroscopy can reach places that were previously inaccessible, such as rocks, to determine if they contain enough gold or other precious metals. Disadvantages: equipment. It is often expensive, making it unfeasible for widespread lab use. Precision. Other chemicals in the sample or atmosphere can interfere and distort the results. Sample type. It can only analyze solutions and requires a large sample. Refractory elements. It has problems with refractory elements. Limited linearity. Flame atomic absorption spectroscopy has limited linearity, limited to one element at a, limited to one element at a time. Flame atomic absorption spectroscopy can only measure one element at a time. So this was all about atomic absorption spectroscopy. Thank you.